Today's my talk is about transcriptional regulatory network for cell type specific gene expression. There are various types of cells in the body of multicellular organisms. Cells in each tissue differentiate from tissue-specific stem cells. For example, all hematopoietic cells differentiate from hematopoietic stem cells, HSC, as shown in this slide. Terminally differentiated cells are derived from HSC through intermediate progenitors, such as multipotent progenitor MPP, common lymphoid progenitor CLP, common myeloid progenitor CMP, and so on. Cells are identified by cell type specific gene expression, including these cell surface markers. Cell type specific gene expression is regulated by a regulatory network of transcription factors. Transcriptional regulatory network plays a critical role in determining cell fate. In this study, I construct a transcriptional regulatory network for cell type specific gene expression and investigate it from the viewpoint of network structure. The expression profiles originally published by Amit's group are reanalyzed to identify cell type specific transcription factors. The expression levels of transcription factors in HSC, CLP, and CMP are compared with those in MPP. HSC specific, CLP specific, and CMP specific transcription factors are defined when their expression levels are larger than those in MPP. Inversely, MPP specific transcription factors are defined when their expression levels are larger than those in HSC. Here, I would like to explain how transcriptional regulatory network is constructed by this toy model. There are 4 transcription factors A, B, C, and D. The expression of transcription factor is regulated by other transcription factors of which binding motif is enriched in its promoter region. For example, transcription factor B is regulated by A that binds to binding motif in promoter region of transcription factor B. B is expressed and then regulate expression of C and D. Not only binding motif of B but also of A is enriched in promoter region of D. So D is regulated by both A and B, and then regulate A to bind to binding motif in promoter region of A. According to this relationship between transcription factors, a regulatory network is constructed as a directional graph. List of transcription factors are given into a Regulon database to detect the enriched transcription factor binding motifs in promoter regions of listed transcription factors. Based on the relationship between transcription factors, transcriptional regulatory network in hematopoietic stem and progenitor cells are obtained as directional graph. These are transcriptional regulatory networks of HSC, MPP, CLP, and CMP. Circles in the network indicate transcription factors, and arrows indicate relationship between transcription factors. Transcription factors in the network are clustered according to their network structure. by Spingler's algorithm. Groups are indicated by colors. HSC network looks dense as compared with other networks. This is HSC network. As you can see, borders between groups are ambiguous in HSC network. On the other hand, the border of groups becomes clear in MPP network. As well as MPP network, borders between groups become clear in CLP network. A few transcription factors are located as hub in the network. For example, CEBP family members are located here in CMP network. CEBP family members are well known as key regulators for differentiation of myeloid cell lineage. These transcription factors with many edges regulate many transcription factors. On the other hand, many transcription factors are regulated by a few key regulators. The distribution of edges is biased. Next, I investigate degree distribution of these networks. These graphs shows degree distribution of HSC, MPP, CLP, and CMP network. Horizontal axis indicate number of edges per node, and vertical axis indicate its frequency. Both axes are log scale. It seems that these distributions follow a power law. This graph shows degree distribution of HSC network. Degree distribution of HSC network is wide and continuous. On the other hand, degree distribution of MPP network is wide but not continuous. This graph shows degree distribution of MPP network. Transcription factors in MPP network are clearly divided into two groups with high and low degree. This graph shows degree distribution of CLP network. Similarly to MPP network, transcription factors in CLP networks are clearly divided into two groups according to degree distribution. This graph shows degree distribution of CMP network. As shown above, transcription factors are clearly divided into two groups. A few transcription factors with many edges in transcriptional regulatory network unidirectionally regulate many transcription factors. To confirm the direction of regulation, diode sensor, that means each relationship between two nodes in the network is investigated. As a result, mutual diode drastically reduced as compared with asymmetrical diode from HSC to progenitors. This result indicates that unidirectional regulation increases through hematopoiesis. This illustration schematically shows two-layered structure of MPP network. Transcription factors with high degree occupying the central region of MPP network mutually regulate their expressions. This is regulatory layer. Transcription factors with low degree surrounding network are mainly regulated by only a few transcription factors. This is output layer. Two-layered structure in transcriptional regulatory network becomes clear during differentiation process. Results of this study are summarized. Transcriptional regulatory networks of hematopoietic stem and presenter cells are estimated. Transcription factors are clearly divided into two groups according to degree distribution. Transcription factors with high degree in regulatory layer mutually regulate their expressions, and transcription factors with low degree in output layer are unidirectionally regulated by a few transcription factors. Modular structure is developed in progenitors by the restriction of mutual regulation between transcription factors. Modular structure of transcriptional regulatory networks are developed prior to terminal differentiation. In progenitor cells such as MPP, CLP, and CMP, two-layered structure is clearly observed. That's all. Thank you for your attention.